See that when you compare responder and non-responder, you start to emerge cytokines such as IL-12, stem cells, growth factor, IL-17, that was significantly different between the two groups. But still, these are not enough to serve as predictor. So in order to have a predictive molecule that could enable us to tease out this group, we use four stepwise variation selection, use a Bayesian information criterion, and so then we find group of cytokines such as IL-12 again across different lineages under lineage 2 and even lineage 6 that were associated with negative effect of the drug. So high level of IL-12 were not enable the patient to control the bacteria, whereas stem cell growth factor and IF7 were associated with good treatment response. So these cytokines are potential biomarker that could enable us to tease out those that are responder or non-responder. So I'll just stop there by concluding, saying that, developing this sensitive method, clinical strains of enable us to the mechanism previously, overlooked, that tyrosine kinase can directly affect the bacteria, and this effect vary with the different lineages, and this end up with a pattern. So drugs such as imatinib were lineage-specific interaction, whereas gefapixant and ilotinib could control all the different lineages that were tested. Monocyte that were used here as a proxy of host immune defenses proof is said to be a useful tool that could be used to tease out people with a different profile, and the identified biomarker can be used in the context of a clinical trial. So clinical trials of host-directed therapeutic should therefore account for both host profile and bacterial diversity, because not every patient might need the same drug or even the same combination or doses. I would like to end by thanking a member of my team in the Gambia and many students that have passed through my lab, and a lot of collaborators, the TB CC passed through my lab, and a lot of collaborators, the TB CC platform that have been in existence for 20 years really enabled me to shape this type of...
Yep. Dr. Bediakon, please.
Thank you very much for a very interesting talk. I just had a question. You showed I think you summarized some data from previous publications where you said individuals with lower cytokine responses than MTB, those with MTB. So I'm just curious, are those studies done in the same population? Are the M. africanum people living close to the MTB? Can you kind of explain that? And then if so, typically in West Africa, what is the ratio of M. africanum versus MTB infection?
Thank you very much for that excellent question. You really allowed me to expand in 15 minutes. It's very difficult to summarize this type of study. So all the study, about 7 publications that are summarized in that diagram were done in the Gambia, where you have M. africanum in the community. So M. africanum causes about 50% of all TB in West Africa. It's an interesting strains of TB, because you can imagine, despite the huge, imagine this trial is being endorsed by WHO. So should we just adopt this type of regimen without, doing some further research? So these are just questions that we need to ponder upon while, we'll move forward.
Dr. Kwashi.
Thank you. I'll sort of stay along the same track, and because in the beginning you talked about the WHO recommendation and looking at your data and other data somewhat related to the talk before, do you think it's a time where this one-size-fits-all sort of pronouncement that this is the gold standard and this is we need to go beyond that? Is more of a comment.
Your statement summarized everything. You can see my bottom-up approach really is against those conventional one-size-fits-all. I think we are very different, and Africa has one of the greatest diversity when it comes to human. Is not because of our color, but the genetic diversity is really huge. Not only the hosts are different, but I'm sure you the bacteria are also different, and the way they respond will be different. So when you come with one single regimen across the continent, not only in Africa, even in other parts, I'm really a very strong advocate of a stratified treatment that takes into account, those regional specificity of the strain that are circulating. At least that we can type and know.
One more question, then we move on.
Yeah, very nice talk. Do you have an idea of the targets for these inhibitors in the mycobacterium and how redundant they are?
So I'm not showing that data. That's something that we're working on. But what at least we have done with whole genome sequencing is to, map out the different kinases in the bacteria. And what I can say to you is that, the abundance and even the mutation that I find in this kinase vary with the strains. Yeah. Okay. But that's really what we're working on.
Thank you.
All right, thank you very much. I think we have to move on. So we will start off with the fellow session, and we have 3 presentations. The fellows have 12 minutes for their presentation, and the first presenter is Edwin Magomer, and he will present on lower T cell activation and elevated plasma beta chemokine levels in HIV-2 and HIV dually infected individuals.
Thank you. So my name is Edwin Magomere, and I'm going to present on low T cell activation and elevated beta chemokine levels in plasma in individuals who are infected with HIV-2 as well as HIV dual infections. Okay, it's not moving. Okay, so we know that currently there's about 40 million people who are HIV infected, and majority of them are residing on the continent of Africa. I don't know, it's not. Okay, sorry. Majority of them reside on the continent of Africa. About 26 million cases are on the continent. But what is very surprising is that when you look at these 26 million cases, you see that only 5 million cases come from West Africa, but the remaining about 20 million come from about east and southern part of the continent. So the question here is that why is it that we have such a disproportionate share of this burden across the continent? And one thing we know about the West African epidemic is that there is co-circulation of both HIV type 1 and as well as HIV type 2. And another fact is that the region is predominated by a circulating recombinant virus. So just quick biology of the virus. The virus is a lentivirus, is in the family of Retroviridie as well, and there are 2 major types of the virus. The first type is HIV type 1, which is grouped into 4 different groups, and the group M is the most predominant group with different subtypes as well as circulating recombinant forms, and type 2 has 8 groups, and only 2 of the groups have been shown to be in circulation. So looking at this map, we see that there is a distribution of HIV type 1 subtypes across the continent, and I want to draw your attention to the West African epidemic, where you see that the circulation is mostly by a recombinant form. Moving forward, we see that HIV type 2 is mostly largely confined to the West African epidemic and has been reported to some other continents, although at a very low frequency. So when we talk about co-circulation of both HIV type 1 as well as HIV type 2, there is a concept of dual infection, which means that individuals are infected with both type 1 and type 2 at the same time. And what happened is that there are studies that have shown that these individuals are likely to have a natural protection against acquiring super reinfection with HIV type 1. And subsequent studies have also looked at how disease progression occurs in these individuals And what has been reported is that those who have HIV type 1 alone tend to progress a bit faster compared to those with HIV dual infections. Again, the question is that what mechanisms underlie this attenuated disease progression phenotype? So studies have implicated T cell memory cells in response in trying to control the dual infection among these individuals. However, what we don't know is whether other aspects of immune responses are also involved. So the rationale of this study is that we know that there is HIV type 1 and it has been extensively studied, and what we know is that the virus usually progresses very faster. It's a faster progressing virus. It also has usually high viral loads, and these individuals have experienced CD4 depletion very fast compared to, it's not working, compared to HIV type 2. And again, we see that in HIV type 2, there is a slow disease progression. There is a very high viral load, very low viral loads, and also we have slow CD4 depletion in HIV type 2 individuals. But what we don't know is really how the disease progression in HIV dual infection is controlled. We have previously performed some studies using cell lines, and we have shown that, okay, when we infect cells with both HIV type 1 and type 2, we see reduced infectivity among these individuals compared to those who have HIV type 1 alone. We also suspect there is contribution of viral genetic factors in this slow disease progression, and also there is possibility of involvement of immune responses. So today we are going to look at how these immune response factors are contributing to slow disease progression in HIV dual infected individuals. And the first objective will be to assess plasma cytokine levels in HIV1 and HIV2, as well as HIV dual infected individuals. We will also characterize T cell immune profiles in HIV1, HIV2, as well as HIV dual infected individuals. So what we did, we collected samples from 6 hospitals in Ghana, and we used a purposive sampling approach where we see that our target population was HIV type 1, HIV type 2, and HIV dual infected individuals. And during our sample collection, we administered a questionnaire as well as retrieved some data from patient records. We went on to collect blood samples from these participants, and when in the lab, we screened for various co-infections as well as confirmed the HIV typing. So here is a summary of the participant characteristics. What we see is that we have a total of about 170 samples, and we saw that there is quite advanced in age, the HIV type 2 group, and we also see that in HIV type dual infection, there is a bit lower viral load compared to HIV type 1 alone. So moving forward, our first objective was to look at the plasma cytokine levels in the plasma of these individuals, and we used a 20-plex luminex assay to do that. So what we saw was that there was a general increase in the levels of beta chemokines in HIV dual infection as well as HIV type 2 infections. And what this data means to us is that usually beta chemokines are natural ligands for CCR5 receptors, which are also required by HIV to infect cells. But when there is high levels of these chemokines in the system, it is possible that the beta chemokines are going to bind to their cognate receptors and exclude the virus from using the same receptor to enter the cells. So we went on to compare viral loads of HIV type 1 between individuals who are HIV-1 mono-infected and those who are HIV dual infected. And you could see that there was a significant difference between them. And then we were asking the question, is it possible that some of the cytokines are correlated with the low viral load we see in HIV dual infections? So what we did some correlation analysis, and we found that the trail as well as MCP1 were correlated, modestly correlated with HIV viral load in dual infections, and IL-17 was positively correlated with HIV viral load in dual infected individuals. So we went on to look at the pro-inflammatory cytokines, and we did not see any difference between the pro-inflammatory cytokine in infected groups as well as the HIV negative groups. And so then we asked the question, if these T cells usually contribute to the production of the cytokines, how is the population of T cells looking like in these individuals, as well as if they are contributing to what we are seeing in plasma. So we performed a flow cytometry assay on these individuals, and what we did is to do a 16-hour simulation with the HIV-specific HIV-specific peptides. We went on to acquire the samples on flow cytometer and then analyze the sample on FlowJo, and this is our gating strategy that was isolating for or helping us to get the characteristics of CD3 cells. So moving forward, what we did is that we analyzed CD4 as well as CD8 cell population in our CD3 cells, positive cells. We analyzed CD4 as well as CD8